Hi, I'm Valentina Pacella, and I'm presenting the exploration of the fabric of the human mind via the brain cognition space. The brain cognition organization, as we know it, is mostly derived from fMRI studies that associated activation during specific tasks to specific cognitive construct. However, if some functions seem to be segregated in the brain, others seem to rely on common brain structure, and this challenges the conception of the brain cognition organization built on activation studies. Moreover, the recursive validation of existing cognitive constructs hinders the exploration of new cognitive functions whose existence is suggested by rare neurological manifestation observed during clinical practice. So the goal of this study is to unveil the latent organization functions in the brain and to provide a quantification of the degree of reliability of functions studied by via fMRI. Moreover, we want to provide a predictive framework to explore functions that have not been described so far. To do so, we leverage onto the meta-analytic approach that allows for less bias and systematic error, and is helpful to combine and summarize the data and potentially offer insights that are not immediately apparent from the individual studies. So we exploited the Neurosynth repository that computes meta-analysis of activation related to specific cognitive terms by scrutinizing the fMRI literature. And we selected 506 terms that correspond to 506 meta-analytic maps. We parceled the maps with a multi-parcellation approach, and we exploited the uniform manifold approximation and projection approach to reduce the dimensionality of our data set while retaining its core structure. So we obtained the embedding of our meta-analytic maps onto 2 dimensions, and we called it the brain cognition space, where maps organize themselves according to their similarities and cluster far away apart according to their differences. We identify in the branches of the space specific cognitive domains, while we were not able to identify any cognitive domain from observing the center of the space. And the position of the branches reflects the integrated anatomical and functional dynamic between the cognitive domains. To compute the degree of reliability of the maps studied via fMRI, and also to see what's going on in the center of the space, we built the rationality index. The logic behind it is to predict the morphology of a territory in a specific coordinate. We leverage on the observation of nearby territories, and it's the same for our space. So to predict a specific map, we will leverage on the similarity with the other maps of the space. However, if the nearby territories are corrupted somehow, this bias will reflect also on our prediction, and it's the same for our space. So if we are relying on maps that have been studied with poor control task, or have been poorly theoretically conceptualized, this bias will reflect also onto our prediction. So we computed Euclidean distances between each and other maps of the space and used these distances as independent variable in as many models as our maps. And our rationality index will be the correlation between the original and the predicted maps. And the color code in this figure shows the rationality index of each map of the space, going from low to high rationality. And if we see a few examples of high rationality maps, such as the listening map here, we can see that we were able to highly predict the activations corresponding to listening, while an example of poorly predicted maps will be consciousness that relies in the center of the space. And we zoom in, we see the example of maps relying on the branches of the space, but I think more interestingly, example of maps relying in the center of the space. And here we see terms such as timing, flexibility or umbrella terms such as executive functions. Moreover, we observe interhemispheric imbalance between the left and right hemisphere in terms of rationality index, with the left that have been more rationally explored. And also, we computed a map of highly characterized structures, so that reflects most rationally explored regions. And the results show the involvement of the inferior frontal gyrus, superior temporal gyrus, presental and postcentral gyrus, parietal and frontal eye field, and subcortical structures. We also compare this map to the 5 resting state gradients that summarize in a data-driven way the brain activation driving human cognition, described by Margulis and colleagues. And we show that our map of most rationally explored region refer to both unimodal and higher order cognition. Finally, we hypothesized that in the gap of the space, we would be able to predict the anatomy of new functions. And to prove this hypothesis, we selected new meta-analytic maps from Neurosynth, and also new raw fMRI results. We parceled them and we projected this new data onto our space, extracting the coordinates and using the coordinate to to compute the prediction of each new map. And as you can see here, we were able to predict with high precision maps that landed onto the branches of the space, as in the example here for meta-analytic map and for raw fMRI data. In conclusion, we provided the brain cognition architecture via the VCS, and we were able to identify which are the maps that have been reliably studied so far and which are the cognitive terms that we should work on more. And also we provided a predictive framework to go on in the exploration of the human mind with old and new cognitive functions.